Welcome everybody. I see there's still people more being entered. For those of you who are in the waiting list, the waiting room, I hope you enjoyed the refreshments that were being offered. For those of you who don't know me or can't read little screen, I'm Janet Warren, and I coordinate the Hamilton Science Faith Forum, which is a Hamilton area chapter of the CSCA. And this is our first and possibly only meeting of the fall. We are, like most people, still going to be virtual, but stay tuned. And if possibly next year, we will likely be meeting again at McMaster Divinity College. If you're not on the Hamilton mailing list, feel free to contact me or Mark McEwen through the CSCA. He manages the mailing lists. If you're not familiar with the CSCA or the ASA, again, check the website. There's lots of information. And Dana might put those in the chat box afterwards as well. So for today's talk, we are welcoming Alan Dickin. Alan's been involved with the Hamilton group for, at least as long as I've been involved, possibly longer, so more than 10 years. And he's a retired or semi-retired emeritus geologist from McMaster. And he's been very interested in the science-faith dialogue, which obviously is what ASA and CSA are all about. So Alan has spoken for us before. He's particularly interested in biblical history, ancient history. He's written a couple of books, scientific commentary on Genesis 1 to 11, and From the Stone Age to Abraham, a biblical history of the ancient world. I'm sure you can contact him if you want more information on those books. But today he's going to talk to us about what can genetics tell us about the origins of Adam and human civilization. So over to you, Alan.
Okay, well, thank you everyone for joining us today. It's great to see you all. And you can see that I have a subtitle for my talk here. Adam and Eve and the PCR test. So I trust you'll find this interesting and thought-provoking. Now, the PCR test has been in the media a lot the last 18 months. But surprisingly, I've never heard anyone really try to explain what it is, and certainly not explain how it connects with collecting of evidence about human evolution. And so what I want to do today is try and join together some of those dots and also join together the scientific story with the story of origins and Genesis. So the PCR test, which, excuse me a sec. Yes. The polymerase chain reaction, the process by which DNA is copied both in nature and the lab. See if my pointer works here. Okay, so on the right here, you can see one step in this process being shown, and we have a template strand along the top, and then we have the strand we're building underneath it. And as it says on the left, polymerase is an enzyme that catalyzes the addition of a new nucleotide with the correct base to the hydroxyl group of the last nucleotide on the chain. So you can see here in this picture, the new nucleotide here is being maneuvered into position. Its base is such that it will match up. The A matches to the T for the double helix, and the polymerase is catalyzing this reaction here, whereby the nucleotide will be attached to the hydroxide group, and so on. Yes, so DNA sequencing is what we're interested in, which is to say determining the order of bases in the DNA, which of course is the language of life. And DNA sequencing is a quantitative chemical process. It doesn't occur by magic. you need about 1 billion copies each piece of DNA in order to sequence it. So the first stage of DNA analysis is to make copies, and you do that by PCR. There are 2 stages. Firstly, you have to heat to denature the DNA, and then you make a copy. And this is illustrated on this information sheet from the NIH, which seems appropriate. Here we have our double helix at the top. We're going to separate it into the 2 separate strands. We're going to start copying with a primer, and then using nucleotides in solution, we're going to build a copy of the DNA until we end up with a new double helix, and then repeat. And if you repeat this 30 times, you should end up with 1 billion or so copies of what you started with. And then we want to sequence the DNA. Sequencing is also done by PCR. The key point is now, though, you interrupt the copying process at random points, adding one of 4 different colored markers to identify the last base added. The result is a lot of DNA fragments at different lengths, which you put in order of size by gel electrophoresis. So we look at the left-hand side here. We have our template, we have our primer, which is going to start copying, and then we start to copy our DNA, but at some random point, this copying process is interrupted, and we're going to attach the fluorescent marker, which is going to identify the last base that was added. Then when we put all of those fragments in order, what we will get is actually a readout of the order of bases in the DNA. Now, you might say, okay, so what's this? What is the bit about when you interrupt the copying process at random points? This is the clever bit. So in your reaction vessel, you put your template, your primer, and you put in a whole bunch of nucleotides with the 4 different bases, but you add 1,000 times less of a special sabotage nucleotide, I'll call it. And the sabotage nucleotide, when it's added, it prevents any further replication of that strand, and it attaches the fluorescent marker. And the guy who came up with this idea, of course, is Fred Sanger, who won 2 Nobel Prizes as a result. All right, so there's a limit to how long a piece of DNA you can sequence, but because you have to put all the strands in order of how long they are, the maximum is a few hundred bases. So if you have a very long piece of DNA, you have to break it into pieces and then sequence each piece. Then you virtually reassemble all these pieces, having sequenced them, based on the overlapping sections which you can match up. So here in the picture is Fred Sanger examining an early electrophoresis image from the late 1970s. Now, this point in the development, they hadn't yet conceived the idea of being able to attach the fluorescent markers. So at the time, the method they had available was to attach a radioactive tag, and this was the same radioactive tag for each of the bases. So what this meant was you had to do the experiment 4 times over, one experiment for each of the sabotage bases, as I called it. Then you would do electrophoresis on each of those 4 experiments, and the result is you get 4 electrophoresis columns here, and where the radioactive tag comes out for each of the sabotage bases, which identified where the base is, then you get a radioactive concentration, and that fogs the film and creates the black band. And so that's the result he's looking at there. And as you can gather, I hope that it was a very laborious process that probably most of us would never even have attempted. Well, your body contains about 100 billion kilometers of DNA, which is approximately, we're told, 15 times the distance to Pluto. A typical cell nucleus is about 5 to 10 micrometers wide, but that nucleus contains 2 meters of DNA. So consequently, it has to be coiled up when it's not in use. We have a coil of coils, supercoils, and then we screw it all up together, and this finally makes what we would see under a microscope as a chromosome. All right, so if we look at human DNA under the microscope, this is a picture of human DNA, and there are 3 fundamental types which we need to consider. So firstly, is what we call autosomal DNA. It's the 22 chromosomes that store most of your genetic information. Then we have down the bottom here, so there's all the 22. The 23rd one, we have the Y chromosome, which is only present in males. It's a kind of filler chromosome if you don't have to have 2 X chromosomes. And finally, we have mitochondrial DNA, which is completely different. Mitochondrial DNA is not in the nucleus, but it's present in mitochondria in the cell. They're the energy-producing organelles in the cell, and their mitochondria is separate, and it's in little rings. Okay, so we want to consider now the relative sizes of each of these types of DNA. We'll come to the picture on the right in a moment. Okay, so firstly, mitochondrial DNA. It has about 16,500 bases, and there's over 100 copies per cell because every mitochondrion has one. Y chromosomal DNA has about 60 million bases, and there's only 1 copy per male cell. Autosomal DNA has over 3 billion bases, and there's a copy in every cell. If you compare that, for example, with the COVID genome, it's RNA, but it amounts to the same thing. It has 30,000 or so bases, and typically 100 copies in an infected cell. So you can see the COVID genome is somewhat comparable with the mitochondrial DNA in its size. Now, on the right-hand side, this graph shows the decreasing cost of sequencing over 30 years from 1980 to 2010. And what you can see is that at the beginning, the late 70s, it cost more than $20 for every base that was determined. Then, firstly, the process of switching from the radioactive tag to the fluorescent dye, and then of mass production, which is to say doing thousands of parallel electrophoresis separations, sped things up by factors of thousands. And then finally, so the first human DNA, of course, cost 3 billion to sequence. But when we sequence someone's DNA for one of these commercial platforms, you don't need every base sequenced. So by only sequencing the important ones, you cut the cost to, say, $100 per person. So that's the sort of history of process. All right, so now we want to get on to mitochondrial inheritance. Now, when a sperm fertilizes an egg, it leaves its tail behind. So only the nucleus is merged. And so the mitochondrial DNA is only inherited from the mother. This is going to simplify the inheritance of mitochondrial DNA and make it simpler to deal with. plus the fact I just explained that mitochondrial DNA is much smaller, this was obviously where people started in terms of studying human evolution. So here is a human mitochondrial DNA tree. Most of you have probably seen this before in some form. The data are mitochondrial DNA from living humans, and the tree shows how closely their mitochondrial DNA is related. Lineages that diverged earlier will have a greater number of genetic differences, mutations. The dates on this tree are based on the assumption of constant mutation rates calibrated against a 6 million year point of divergence between humans and chimps based on fossil evidence. So now we're going to consider what this data show, the results. So if we look at the right-hand side, wherever my cursor's gone, people from all continents outside Africa on the right are more closely related to each other, which is to say they're on these small sub-branches of the tree, compared with people from Africa, which form the root of the tree, the base of the tree. S is Asia, M is America, E is Europe, U is Australia, P is Pacific. So this is the rest of the world compared with Africa. So what this shows is that modern humans evolved in Africa, because that's where the root of the tree is, and they left Africa sometime 60 to 95,000 years ago. Mitochondrial Eve, who should be in scare quotes, is the last common ancestor of all human mitochondrial DNA. So, question, what has this got to do with biblical Eve? Short answer, nothing. If biblical Eve was the same as mitochondrial Eve, then biblical Eve would have had to have lived in Africa 150,000 years ago. If this was really true, why is the story of Adam and Eve set in Mesopotamia? And how was it passed down for 150,000 years? No, this is not credible. What we should say is Adam and Eve are not the biological parents of the human race, but the spiritual parents of the human race. Now, if we go to Genesis, it tells us about where this happened. The Lord God planted a garden in the east in Eden, and there he put the man he had formed. A river watering the garden flowed from Eden. It had 4 head streams. It goes through the first 2, and then the third is the Tigris, which runs along the east side of Asher, and the 4th is the Euphrates. So what this is saying is that the Garden of Eden was between the Tigris and the Euphrates, which is to say Mesopotamia. Now, what is the temporal setting of Genesis? Well, we read that Adam lay with his wife Eve, and she conceived and gave birth to Cain. Later, she gave birth to his brother Abel. Now, Abel kept flocks and Cain worked the soil. This text shows that Cain and Abel, and by inference their parents, lived after the agricultural revolution, which is around 8000 BC. So then we continue. Cain was building a city and he named it after his son Enoch. The agricultural revolution allowed large numbers of people to live together in cities. Civilization means life in cities. So what we learn from this is that Genesis is describing the origins of human civilization, not the origins of the human race as a whole. So what about the origins of agriculture? From bones in ancient settlements, the domestication of sheep and goats began in the northern Fertile Crescent around 9000 BC, and then it spread out to the east and the west and the south. The origin of wheat occurred in the same place, a place called Karasa Dag, but I don't have time to go into the evidence just now. Okay, so what I want to do now is switch to examining the Y chromosomal evidence for a second line of approach. Now, on the right-hand side here is a Y chromosomal tree. It's very similar to the mitochondrial DNA tree. And this tree can be used to create the diversification plot on the left-hand side. For example, if you look to see where you had a number of branching events, which is to say genetic diversification events, then that corresponds on the left-hand side to a sudden increase in the effective population size, which is basically the diversity of Y chromosomal DNA. And what you can see is that this first event at 50,000 years, this is equated with the out of Africa migration, when human populations moved out of Africa and then spread over the rest of the world. But now we can see a second event between 8,000 and 4,000 years ago, when there's a sudden drop in effective population size, which is modeled, and then a much more sharp increase again around 4,000 BC. This second phenomenon here is attributed to the migration of the agriculturalists after the agricultural revolution. Basically, the farmers wiped out the earlier hunter-gatherers and then they replaced them. Now, what we want to do finally, we've looked at mitochondrial and the Y chromosome evidence. So now we want to look at the complete genomic evidence. That is to say, the autosomal 22 chromosomes. Obviously, we can't look at 3 billion bases. So what we do is we focus on a few thousand locations which have significant variation. They're called single nucleotide polymorphisms, SNPs. When we want, it's still too much data to manage. So what we do then is we use the statistical process of principal component analysis, PCA, to reveal the greatest trends within the data set. And you can see here on the plot here shows the first and the second principal components which have been derived, eigenvectors. And these are the ones that show the greatest spread in the data. For example, here you can see the Bedouin, the Basque, the Lithuanians, the Chechens. These represent relatively isolated populations that are at the extremes of the distribution, and then like everyone else is in the middle. Now, this is modern autosomal DNA, but in order to test the migration after the agricultural revolution, what we need to do is analyze ancient human DNA. This is a whole new ball game in terms of technical difficulty. For one thing, DNA decomposes over time. Secondly, there's a strong tendency to contamination with modern human DNA and also environmental DNA. So this method has only been successful over the last 10 years. But this is the method that gives us conclusive evidence for the human migration of the agriculturalists. Okay, so what you see in this plot, this is the same, this is the same principal component analysis as the previous plot. And in this plot, the black fields around the edges are pre-Neolithic hunter-gatherers, so before agriculture. So for example, we have Southwest Europeans, we have the Natufians, that's in southern Israel, and then we have the Caucasus Mountains. And then finally, we have another group in the middle, which is Anatolia, and Anatolia is essentially where agriculture was first developed, or at least it was right next to Anatolia. And it's the first place where the farmers started to to rapidly multiply. And so what you then see is, if you look at the white and the gray fields, you see how those agriculturalists spread over the rest of the world. So we have a trend, the pointer's being annoying, but we have a trend then to the southwest of Europe through Greece, of mixing trend. We have another trend that goes to the south into the Levant. And we have another trend that goes to the east in Iran. And then finally, we have a trend, which is, I haven't shown here, goes towards the Caucasus and the Russian steppe, and eventually into Northern Europe. In comparison, Genesis describes the spread of the nations from Mesopotamia after the flood due to the sons of Noah. As it says, this is the account of Shem, Ham, and Japheth, Noah's sons. From these, the nations spread out over the earth after the flood, which was also in the Neolithic. So we can see in this plot shows the genetic evidence and how well it ties in with the biblical table of nations, as it's called. So starting in Mesopotamia, we have the movement to the west, which is the Greeks. We have the movement to the north, which is other Japhites, as they're called. We have the movement to the south, which is the Canaanites and the Egyptians. And then we have the movement to the east, which is the Elamites and the Persians. So you can see here a good correlation between genetic and the biblical evidence for the spread of people from Mesopotamia. But having said that, Genesis has different objectives from the scientific objective in its account of history. The biblical story of origins is not concerned with the nuts and bolts of genetics or history. It's concerned with the relationship between God and mankind. If God actually exists, then this must be a personal relationship between us as individuals and God, and therefore it must start with one individual. And Genesis says that after God made the man from dust, God placed him in the Garden of Eden. So the making of man from dust is purely symbolic of mortality. It's not speaking that he was actually made from a heap of dirt, literally. Eden, where the garden was located, is simply Eden. It's simply the name of the plain of Mesopotamia. So the Garden of Eden was a spiritual sanctuary in Mesopotamia. The act of placing the man, which is to say Adam simply means the man. The act of placing the man in the garden sanctuary was the point when an evolved hominid was born spiritually. So after being placed in the garden sanctuary, there, Adam, the man, experienced the presence of God. This life-giving communion with God is symbolized as the tree of life. But the question arises, were Adam and Eve intended to live forever in Eden if they hadn't sinned? And surprisingly, Calvin says no. Referring to Genesis 2, Calvin says about Adam, his earthly life truly would have been temporal, yet he would have passed into heaven without death and without injury. And commenting on Genesis 3, Calvin says, truly the first man would have passed to a better life had he remained upright, but there would have been no separation of the soul from the body, no corruption, no kind of destruction, and in short, no violent change. So what Calvin is concluding here is that mankind was never intended to live forever on earth. He was intended to serve God as priests in God's sanctuary, cosmic sanctuary, before passing to heaven. So in that case, what does the fall mean? We know Adam and Eve sinned by seeking independence from God. The fruit is purely symbolic. They didn't die physically the same day they ate the fruit, the symbolic fruit, but they died spiritually when they were cut off from the presence of God. What Adam and Eve lost was not immortality on earth, which was never possible, because life on earth has always been based on death, which is to say, when you eat plant cells, they die just like animals die. What was lost in the fall was the hope of eternal life. So when Adam and Eve sinned, they lost the hope of eternal life. And we know that because in the Old Testament, there's almost no hope of heaven. So, for example, Psalm 115 is typical. The dead do not praise the Lord, nor do any that go down into silence. So the fall is a loss of the hope of eternal life. But now, of course, the second Adam, who is Jesus, has recovered this hope for us and for all of humanity. So what can we say in conclusion? The genetic evidence shows that humans originated in Africa, but civilization originated in Mesopotamia. Genesis describes the spread of civilization after the agricultural revolution. But Genesis is concerned with Adam as the spiritual father of humanity. Adam was never intended to have earthly immortality, but to serve God on earth before passing to his heavenly reward. We know that Adam failed in his mission, but the second Adam has recovered his calling for us, and therefore he offers us the same opportunities, essentially, that Adam had to serve him on the earth, which is the cosmic temple. And a whole bunch more of the details of this story are in the book that Janet's already mentioned. So thank you very much for listening, and hope I can answer your questions.
Thank you, Alan. That was very interesting.
Just curiosity, Alan, I'm halfway through your book. Thank you for giving me a copy. I'm really enjoying it. You explain quite clearly, of course, that the focus of biblical history is around Mesopotamia and spreading out from there. Just a bit of a kind of cross-check as to a kind of timescale for when people traveled from Mesopotamia right through Asia and crossed to North America. Because I thought that was about 10 or 12,000 years ago. And so those people presumably were not the agriculturalists.
Well, yes, that's for sure. Yes. I mean, that the migration around the whole earth, which began with the out of Africa migration, then later into Europe, the Far East, and then from then into America. So that's essentially the migration of the hunter-gatherers. The point of the evidence from the Middle East is it's showing essentially that there was a second outward migration, which is probably not as well understood or known about as, everyone knows about out of Africa, but the migration that began in northern Mesopotamia and spread out all over, well, certainly all over Europe and the Middle East is less well known. And it's this second migration which relatively closely ties in with the account of the spread of humanity after the flood. And this, of course, depends on understanding the flood happened earlier than many people think in the Neolithic. I could say more, but
I think you've got a question in the chat, and I think Gordon Murray had his hand up as well.
Yes, I was just going to say.
Sorry, Janet.
Yeah, go ahead with Gordon.
All right. First of all, Alan, thanks very much for your talk. As a geneticist, I appreciate it very much. And also, I've read your book, which was extremely interesting for me. But I do have a little question about your book. On page 6, I think you misquote Genesis 4:24, because when I read Genesis 4:24, it doesn't say what you say it says. I wonder if you could clarify that.
I don't know if I can clarify it on the spot, because
you say that Genesis 4:24 refers to people having worshipped Yahweh for I forget to quote, for a long time. But Genesis 4:24 talks about forgiving till 7 times 7, so I'm a bit confused. But you don't have to answer now. You can respond to me by email or in the chat if you wish.
I had a Bible here, but I seem to have lost it. Okay, hang on.
If Cain is avenged 7 times, then Lamech 77 times. That's
okay. Well, maybe it's the number. I'm maybe got the number of the verse wrong. Yeah. Okay, I mean 26. Oops. Yeah, so it's verse 26 is what I'm talking about there.
Okay, thanks very much. Sorry for the sideline.
All right.
Okay, there was a question about genetic analysis on humans in the whole world to see how migration happened.
Well, yes. Okay. I mean, I think you're asking about the out of Africa migration, which the out of Africa migration, the evidence I showed you there was a model of the timing of migration based on Y chromosomal DNA, but it's been verified by fossil remains, that show when people were appearing in, like, for example, southern India, when they appeared in Australia, all this stuff. So, in other words, it's not just one line of evidence here. The out of Africa migration is pretty solid in terms of the last major migration from Africa that gave rise to basically all modern humans. What was the question? I mean, yes, there's a bunch of evidence for that. What I'm focusing on here, though, specifically, is the out of Mesopotamia migration. And the point is, that's the one that ties in with the biblical story of origins. So I think that's best place to leave it.
Daniel has a question, and then Russell.
Yeah, thanks, Alan. I guess I have more of a theological question, so maybe you don't want to delve into this too much, but I was quite intrigued by your point about the mitochondrial Eve not being the same as the biblical Eve, and that makes complete sense to me. However, when you said that the conclusion, therefore, would be that Adam and Eve are not biological ancestors of human beings, but spiritual. Well, that's fine as well, genetically, but I think people will have a very difficult time then explaining, well, what about all the people, Homo sapiens, prior to Adam and Eve? How did they fit into the relationship with God? How are they recognized as being created in God's image or not? And as well as all of those who are around Adam and Eve, but not yet in any kind of, relationship with them. So there's some quite difficult theological issues of how that, the sense of image of God, the sense of the cultural mandate given by God to Adam and Eve. How did that then impact all the rest of humanity?
Yes, okay. Well, as you say, complicated questions. I think I would answer by saying primarily those are philosophical questions. They are questions like, should everyone have a fair chance of hearing God's word, if you like? And I mean, sadly, the truth is, well, it didn't quite work out like that. Not everyone has heard the gospel. I would maybe suggest that the revelation to Adam kind of worked a bit like that. I mean, God had a plan that he started with a person in the Middle East. And, maybe Adam was the first person who was ready for the revelation, whereas other people weren't. So we could, there's a lot of philosophical questions in there. It would take quite a long time to sort of wrestle with some of those. Yeah.
That would be a different kind of book, I'm sure.
As a theologian, I can offer some comments. I think you make a good point, both of you, is that with none of this can we be certain, right? So I think that's important. So one approach is to view the first few chapters of Genesis as strictly symbolic and not related to any, first human at all. A second approach would be to view that story as describing the origins of the Jewish people, God's chosen people, as opposed to the entire human race. Third option would be to say that human, sort of what Alan was saying, but human consciousness only evolved at that point, which might be a little bit late in history, but it's an option. So those would be the three sort of approaches that we could have, if that helps.
Well, I guess I would sort of opt for something in between those. For example, I would look at what we call the Table of Nations, which is the kind of schematic family tree of Noah's descendants, and the ancestors of the Hebrews, Eber, is kind of slotted in there as a sort of almost an adopted son of Noah's descendants, So there's some complications in how Abraham inherited the lineage from Noah. Of course, this is assuming you think Noah was a real person. I think there's good evidence that he was a real person, that there was a real flood, and that there's a huge amount of history in there which we can explore. But of course, we didn't have time to go into it today.
Right. So another point you bring up, it also depends on our views on the relationship between science and Christianity. Because if you take the view that they're separate, and you were a little inconsistent, because Alan, I think you rightly said that the point of Genesis is not really to describe human origins. It's about God's relationship with people. And then at another point, you said that some of the ancient history, etc., especially around civilization, is compatible with the biblical story. And I think that's a key word. I think there's a big difference if we say that the Bible is actually describing some scientific facts that we know versus saying that the biblical story is compatible with certain things that say. So you sort of said we're saying both of those things. My preference would be the word compatible. But anyway, if we should move on, so Russell and then Gordon.
This may be more appropriate for another group, I don't know, but I'm going to throw something out. I wonder if the Adam and Eve story is dealing with the origin of the Jewish people, not of all mankind. When Cain killed Adam and he was thrown out of the garden, and he's thrown out and put a curse by God, he says, well, they're going to kill me. And God says, no, I'll put a mark on you. Fine. Well, who are they? Other people. Now, the biblical literates want to say, well, it's because they don't say in the Bible, but because they had sex with their sisters. I really don't think God is going to have incest is one of his early teachings there. And it wasn't explained in the Bible for a good reason why. The people who compiled it in a long oral tradition before that knew the fact that the people who had killed Cain weren't the brothers and sisters of Adam and Eve. They were there. And I think it's really consistent with the ancient world, because even American Indians saw other tribes sometimes it's not human. And you have a tendency for people to think of their group real tight and not of the all of mankind. All of mankind really is a post-World War II idea. Even the Nazis had the Slavs as being subhuman.
So yeah, I did mention that as one of the possible interpretations.
Yeah, I think it's
one of the possible ways to overcome the discrepancies between what the Bible appears to say and what scientific findings seem to say.
I think something else at work too, and throughout all scriptural interpretation, I think to raise another principle: God speaks people's language, including their images and their previous experiences. And so, like we'd like to know the story. There's all kind of evidence of outburst floods in the last ice age, and those were used to get a point across. And people keep missing the point, and that is, there's only so much God's going to put up with before He's going to kick the snot out of you. Something we need to always keep seriously and don't diss God. Anyway.
Alan, did you have any other comments?
Okay, well, what I would say is that there's a lot more history in here than is usually appreciated. Okay, so the flood, the evidence is the flood was much earlier than most people think, 5,500 BC. There's no evidence that Noah was an Israelite or a Jew or anything, or a Hebrew. I mean, so in other words, the Bible is summarizing thousands of years of history. Actually, before Abraham, there's more history before Abraham than there is in the whole of subsequent human history. So there's a lot more happened in there than most people are aware of. And the Sumerians, essentially, the Sumerians are in there, and they were once God's chosen people. That's what I think the evidence shows. And only later did Abraham become God's chosen people when, basically the Sumerians screwed up. So yeah, there's a lot of history in there, and I think it's all to be explored.
And yes, and lots of layers of meaning. So I love Genesis. Anyway, Gordon?
Thank you. I understand that evidence from Australia indicates that the Aboriginal peoples have been there for a long, long time, maybe 50, 60,000 years. If that's the case, by the timelines you've presented, they would have bypassed Adam and Eve, and yet they are fully human and capable of faith in God.
Right. Well, the point, of course, is that, the Australian Aboriginals, if we can call them that, are part of the out of Africa migration as of 50, 60,000 years ago, and that they are part of the human race. And, as such, they have the same mental capacity, the same spiritual capacity as everyone else. But the story of the gospel, if you like, which is to say the first gospel, which is the gospel of Adam rather than the gospel of Jesus. The first gospel was meant to work in a similar way. It was meant to carry the story of God's love of humanity around the whole earth, starting in Mesopotamia. So something we need to point out here is that when it says in Genesis chapter 1 that all humans have made an image of God, that account is almost certainly a separate account from the story of Adam and Eve in Genesis 2. And so, they're looking at humanity from slightly different points of view. All of humanity has the same capacity for relationship with God, but all of humanity has to hear the gospel transmitted from other humans.
Bob?
Just to confirm things then, Alan, and thank you very much. Is your thinking like others that Noah's flood was restricted in its extent, but still an important sort of foundation for a very critical theological story.
Yeah, well, we have to be very careful about our perspective. People talk about worldwide floods or global floods, I guess. It's a question of what was the known extent of the world in ancient times. If we understand that the flood was 5,500 BC, this is before the development of trade links span the Middle East. They were starting to enlarge, but they did not span the whole Middle East. So it's a question of what was the ancient perspective on the extent of the earth. If you lived near a city of Ur in ancient Mesopotamia, you could not see a mountain from where you lived. As far as you were concerned, the earth was flat as far as it went in every direction. So, you have to look at the worldwide flood from the ancient perspective. And then you would see that, Mesopotamia basically appears to go on forever. It's actually, dead flat for hundreds of miles in every direction. So it's not that it was not that they knew it was a local flood, as John Walton has said, and other people have said, well, if they knew it was local, why didn't they just walk away? That was not God's plan. God's plan was that Noah built an ark, although not like most people think it was. But so yes, it covered the whole known world, I think, is the key understanding, that it wasn't just some minor river burst.
Yeah, thanks. Some might recollect that Hugh Ross at this summer's ASA annual meeting, From Reasons to Believe, gave a talk essentially supporting what you're saying in terms of the local situation and how the Bible uses the word worldwide in many different ways and contexts. So thanks, Alan. It's good to hear that from a geologist.
Well, okay, talking about geology, yes. The geology of, we're talking here about the post-glacial geology of Iraq, and it's actually all up for play, for grabs. There have only been a couple of dozen deep boreholes. When I say deep, boreholes of suitable depth, which would be about 15 meters, sunk in Iraq, that might tell us about when the flood was, what it was like, And one of them was just published 2 or 3 years ago, some boreholes in Uruk, the city of Uruk, which show where the flood horizon might be before the earliest human deposits in Uruk and in Iraq in general. So I think there's a huge amount of research still to be done here. Hopefully it will confirm my story.
Thanks.
Well, thank you very much, Alan, and thank you everybody for joining us on this Friday afternoon. And we'll look forward to seeing you again.
Thank you so much, and goodbye. Thank you.